Assalamualaikum and a very good day to everyone. In this part of our lecture, I will be presenting two types of receptor, which are ion channel link receptor and enzyme link receptors. But first, I will discuss about the first part. In ion channel link receptor, we are going to look into three receptors, which are nicotinic acetylcholine receptor, GABA receptor type A, gamma aminobutyric receptor, and 5-HT3 receptor. The first two will be discussed in detail in the next slide. So I will first discuss the 5-HT3 receptors. This receptor is a ligand-gated ion channel that is associated with 5-HT, 5-hydroxytryptamine, or serotonin. This receptor will form a channel that is permeable for sodium and calcium ion. Upon binding with the ligand 5-HT, it will be activated and induce a rapid depolarization. For the brief explanation for the first two, which is NACHR and GABA receptor, you can give it a read right now on the slide. Let us now go to the next receptor, which is NACHR. Please note that this receptor is made up of two alpha subunit, one beta subunit, one gamma subunit, and one delta subunit. The ligand or neurotransmitter for this receptor is acetylcholine. Acetylcholine will bind on the alpha subunit, and a conformational change will occur that results in the transient opening of the central aqueous channel through which sodium ions penetrate the extracellular fluid into the cell down its concentration gradient. It will produce a localized excitatory postsynaptic potential, which is a depolarization. The voltage change then will open the calcium channels from the calcium ion store, and the calcium ion will bind to inhibitory protein complex of tropomyosin and troponin. Thus, skeletal muscles will contract. Now, we are going to look into enzyme link receptors. Look at the shape of this receptor. It is made up of two domains, which is ligand binding domain and also the enzyme domain, also known as kinase domain. Also note that a single enzyme link receptor cannot work on its own and must go through dimerization in order to be activated and also to work. The kinase domain can phosphorylate residues located on the other receptor chain. It's called autophosphorylation, or we can also look at it as a cross phosphorylation. We also have the receptors with intrinsic enzymatic activity except for tyrosine phosphatases, and it's already been represented in the active state. Let's look at the picture. I'm sorry for the lack of clarity, but this is basically how it looks like. This is RTKs, receptor tyrosine kinases. This is receptor serine threonine, and this is receptor tyrosine phosphatases, and this is receptor guanylyl cyclases. This is the receptors with intrinsic activity. You guys can give this a read. And note that why it is called kinases. Kinases is enzyme that catalyze phosphorylation to activate a protein or an amino acid. In this case, tyrosine, threonine, and serine is the amino acid, the target amino acid that is being targeted by this kinase to go through phosphorylation. Now let's go and look at this important point, which is phosphorylation. We know that proteins can be activated or inactivated in a variety of ways. However, one of the most common tricks is through phosphorylation. Firstly, we must know that phosphate groups will be typically linked on one of the three amino acids that have hydroxyl group, OH, in their side chains. Like we said earlier, tyrosine, threonine, and serine. So the transfer of the phosphate group, yes, is catalyzed by kinase enzymes. And in general, phosphorylation is not permanent. We can flip back the proteins to its non-phosphorylated state through the usage of an enzyme called phosphatase. It's a familiar name because we heard it before, the tyrosine phosphatases, right? Alright, done with that part. Let's talk about one of the enzyme link receptor, which is RTK. This is an example. Let's use the growth factor as the ligand for this receptor. Alright, let's look at this. This receptor will shift from inactive monomeric state, as you can see here, monomeric state, to an active state when they are dimerized. This is followed by the auto-phosphorylation with the cross-phosphorylating that we said just now. And then a binding of the SH2 domain protein, GRB2, will also happen. It is an adapter protein. And depending on the receptor subtype, it will allow the phosphorylated receptor to activate other functional proteins, which eventually will result in stimulation of gene transcription. As you can see here, a lot more things will happen. A kinase cascade will happen before gene transcription will happen, but I will only explain until this part where the RAS will be activated right after this. Alright? This is the component of enzyme link receptors, components and things that are related in this pathway. One is GRB2, growth factor receptor bound protein. GRB2 is an adapter protein that we've seen just now, right here. This is it. This is the SH2 protein. Then we have SOS, son of sevenless. This is a GEF, a guanine exchange factor, and is very much required to activate the tyrosine kinases. And then we have P21 RAS as a small GTPase that is functioning as a molecular switch. I would say that P21 RAS is about right here. Alright, and then we have RAF1 kinase, a type of proto-oncogene for serine threonine. Next, let us discuss how RTKs activate RAS. Firstly, the signal molecule will bind on the receptor domain, and after that, auto-phosphorylation will happen at the amino acid residues at the enzyme domain. As we can see here now, the amino acid is phosphorylated. Then here comes the adapter protein. If we are speaking in the context of growth factor just now, the adapter protein will be GRB2. And then a RAS activating protein will bind on the adapter protein. When we talk about RAS activating protein, it's mostly GEF, guanine nucleotide exchange factor, and in the context that we learned just now, it's SOS, son of sevenless. What is the role of SOS? SOS will activate the RAS protein. How? The SOS will eliminate the tightly bound RAS protein with GDP and exchange it with GTP, thus making the RAS protein activated. This is only half of the part of the transmission signal. There will be more steps on this after the RAS is activated. However, that part will be explained more by Najia in her part. Thank you everyone.